Mark, you had your question I wanted to get to.
T regulatory cells are known to be site of persistence in HIV, and likely those cells are going to be more resistant to cell death. So is there any more thoughts on what direction the Tregs are going to play in the role of cure research?
Great question. Who wants to try and respond first? Please come off mute.
It is a great question. I wish I knew more. I should know more on the topic, but I don't know so much.
Yeah, as you know, Mark, the Treg area at the beginning was, there was a lot of hope that there was a real relationship between viremia or control or immune response and the levels of Tregs, because Tregs can go up with an infection status. But a lot of that data didn't really provide a clear correlate regarding the direction and the frequency by which the amount of control or immune response could be predicted by the levels of Treg in a person. Now, I think there is still a lot of interest in releasing the inhibition, but most of that interest has actually centered around checkpoint inhibitors, as opposed to Tregs, kind of like releasing the negative signals that may be coming directly to a T cell. And there was even a presentation still this year about anti-PD-1, and I know that there are ongoing work on other negative checkpoints as well. So I would say that if you think about the Tregs as a negative regulation on the immune response, that what we're seeing more is the trying to use these anti-cancer strategies that have developed antibodies to target specific molecules more than targeting an entire cell.
Yeah, but if the T regulatory cells themselves can get infected and basically be perhaps resistant to apoptosis, would that present a greater challenge to a cure strategy?
Yeah, I mean, I just don't know the data that supports that a Treg would be resistant to apoptosis and enrich itself in the presence of viral replication. Because you're suggesting that a cell can be infected, be activated, and it's not going to be expressing the virus nor dying. And so it would basically expand itself preferentially while at the same time being infected. I mean, we know about the clonal expansion and that it can occur, but there's no data linking clonal expansion to Treg phenotypes directly, meaning that every time we see a clonal expansion is a Treg. So that part, I think we don't necessarily, I don't know of any data that can say, okay, well, the Treg is responsible for immunosuppression based on the expansion of infected cells that don't die. That I'm not aware of.
Okay. And Mark Milano, sorry for the mess up, but did you want to ask your question now?
Sure, thanks. I just wanted to ask if there was any, has been any development at CROI or anywhere else on a problem that comes up in my HIV workshops over and over and over again. And that is that subpopulation of people who have been undetectable for years and cannot get their CD4 counts above 200. We always say just wait and wait, but is there any movement on raising CD4 counts when you're chronically undetectable? And any hope for these people who cannot? It's really tough because they're at risk for all these OIs, even though they've been virally suppressed for years. Are we seeing any talk about that? What can they do?
Great question. Thanks, Mark. Any of our panelists want to chime in and respond here? And then Michael posted in the chat, immunologic non-responders is what Mark is referring to in his question.
And I thought in Case Western, they were actually kind of looking at gene therapy approach to help with that, since they were seeing some sort of benefit from the early Sangamo trials, but I don't know if that trial is currently ongoing. Richard, do you know?
There's long-term, this is Matt Sharp, there's long-term follow-up in the early Sangamo trial, but the company refused to do any further research.
Hmm.
There was a the NIH had given some money to look at another trial, a second trial, but I think that trial, because of COVID, just completely died.
Okay.
But that was it.
And Simon posted in the chat about from some fostemsavir upcoming studies for low CD4 folks.
And I think there might be one remaining Sangamo study. I'm not 100% sure if but it was listed as still ongoing in Cleveland, but it wasn't really focused on people with low CD4 counts. And it is an area where there hasn't really been, there was some activist sort of activity trying to encourage development of adjunct therapies. That wasn't really all that successful. IL-7 was studied for a while, but then the company developing it went bankrupt. There was a study from the FDA at CROI last year trying to see if there was kind of endpoints that could be used as surrogate endpoints for this type of treatment study, and they didn't really have much success. So really to prove a benefit, you'd have to do very large trials that would look at clinical endpoints. And so it's a difficult area to encourage industry to engage in. There was just a meta-analysis of studies published, which found, kind of as we know, that nobody's really seen any particular effect with anything. And I can post a link to that in the chat. But it's something that we're definitely keeping an eye on and still trying to encourage more studies of.
Bob Buckley's just reminded us the name of the study is Trailblazer. I knew it had a name. Thank you for that. And he says that it is restarted. So, and it's, but it's not looking at folks with compromised immune systems. So thanks for that update.